We are going to be going over some of the fundamentals of the digestive system. Specifically, we want to focus in this lecture on the anatomy and the physiology of the stomach, the liver, the gallbladder, the pancreas, and the duodenum. But let's start with how digestion begins. We do have two basic types of digestion. One is called mechanical, and the other one is known as chemical. So if we take, for example, the location where digestion begins, the mouth, we have mastication, which is chewing, as our mechanical type of digestion. And we have digestive enzymes through saliva as our chemical type of digestion. For example, salivary amylase. Or also an example is lingual lipase, right? So salivary amylase for the breakdown of carbohydrates, lingual lipase on the other hand for the breakdown of fats. The mechanical digestion, which uses teeth, and you can think about chewing as mastication, uses a physical breakdown of food. Other types of mechanical digestion that we can see, for example, in the stomach is gastric churning, and that's using the muscles of the stomach. Gastric churning is another type of mechanical digestion which takes place in the stomach. Now, this particular lecture is going to focus on this part of the anatomy. And we're going to see how these organs in that window all play a role in working together in breaking down the food. One thing you do want to notice is that you have the stomach that is somewhat in front of the pancreas. So you do have to remove that stomach in order to see the pancreas in its entirety, which is being shown in this picture right here, for example. There is the pancreas. You can also see that the gallbladder has ducts or tubes that lead towards the pancreas. And the pancreas has ducts as well. And we're going to see how the ducts of the bile coming out of the gallbladder and the ducts of the pancreas are linked in order to send their secretions out, their exocrine secretions out, into the duodenum, or that first C-shaped part of that small intestine. So, what happens when you first put food in your mouth is that the chemical digestion is occurring from the saliva coming out of the salivary glands. At the same time, you have mechanical digestion from the mastication. And when food is mixed with the saliva, you get a semi-solid bolus, which then has to be transferred to the oropharynx behind the mouth where swallowing or degglutination is initiated. As the bolus travels down the esophagus, it eventually finds its way into the stomach, and the stomach is a location where we have further chemical digestion using gastric juice and the enzymes within gastric juice, plus the gastric churning of the mechanical digestion that takes place there. So let's go ahead and look at how the liver, the gallbladder, and the pancreas are linked. The first thing to notice and to realize is that the liver is the site of bile production. And the gallbladder, on the other hand, is the site of bile storage. Now, bile is an emulsifier of fats. And this just means that it takes fats and it breaks them down and allows for them to become water soluble. This is the same way that soap works when it takes grease off of your hands. So you want to think of bile as a secretion that is important for foods that are high in fat. And the idea would be that in order to break up that fat, you would need a type of emulsifier or soap to break up that fat. If you look at a close-up view of the ducts coming out of the liver, you notice that there is a left hepatic duct and a right hepatic duct. So imagine that the bile is being made in the liver, and then that bile starts to get sent through these ducts or these tubes. Notice that they're joined together here by a common hepatic duct. And so now that bile is coming down this way, and then finally it goes into a common bile duct. However, we should note that there are sphincters that close off that common bile duct further on down. And these sphincters are closed when we are not eating fatty foods and we don't need that bile, which means that bile starts to back up and to fill in that common bile duct. And when it overfills, it starts to fill into this branch known as the cystic duct. And notice where the cystic duct is leading to. It's leading to the gallbladder. So the gallbladder, therefore, and you can see how it's shaped like a bulb, is a storage area for bile that we're not using at that moment. The gallbladder is muscular, so that when we do need to use that bile, it can squeeze on that bile and push that bile through that cystic duct all the way down that common bile duct. And if the sphincter is open, then it can go through this opening at the bottom here, which we will discuss later on, and make its way into the duodenum, right in there. Notice, however, that not only do we have a secretion coming out of the common bile duct, we also have another duct that is associated with the pancreas. And this duct, known as the pancreatic duct, if you do follow it, you will see that it meets that common bile duct in a structure known as the ampulla of Vater, which is basically just an enlargement where two ducts meet. The idea being that the secretions from the pancreas and the bile get to mix together in order to make one secretion which can come out of that area and meet whatever broken down food is moving down that duodenum. Here we can see some further anatomy that can help us understand this a little bit, which is that, for example, if we follow these ducts again, we've got the right hepatic duct, the left hepatic duct, the common hepatic duct, leading to the bile duct. And the cystic duct is the one that is coming out of that gallbladder. From the pancreas, we see that we have that main pancreatic duct going right down the middle of that pancreas. And if we follow that, now here you'd have to kind of go behind this to follow it, but this is the continuation of it, of that pancreatic duct. And there where the two ducts meet is the ampulla of Vater, as was discussed previously. And notice that in the duodenum, you have this nipple-shaped structure known as the major duodenal papilla. And you can see that, in fact, it's raised, and there's a hole in the middle of it. And that hole is, of course, where the secretion would come out and find its way into the duodenum. Now remember that the duodenum is the first part of the small intestine, which means it is receiving the chyme. That's what we call it. The digested material from the stomach is called chyme. It's receiving the chyme from the stomach that's coming down into the duodenum. Another structure to notice here, which we'll see again later on, is that you also have an accessory pancreatic duct in some individuals, which is a smaller duct of the pancreas. It is not associated with the common bile duct. It's on its own, and therefore its papilla is smaller, and it's named the minor duodenal papilla. Let's review some of the basics of the pancreas to understand what is flowing through that pancreatic duct. We did go over the histology of the pancreas in the endocrine chapter. So if you need a review of that material, please look at endocrinology for a discussion on what the histology of the pancreas looks like. Right now, we'll be using this image to help us. We're taking a little square right in here, and we're zooming into that square in order to see what this pancreatic tissue looks like. So this should be a review. Within that square, we will notice that you have both an endocrine and an exocrine portion to the pancreas. The endocrine portion is known as the islet of Langerhans, or the pancreatic islet, which is being depicted here in this light blue. That's where you will find cells that make insulin, as well as its counterpart glucagon, and that, of course, is for blood sugar levels and the homeostasis of blood sugar levels. But today our conversation deals primarily with instead the exocrine portion of the pancreas. And that's coming out of these pink structures known as acini. And you can see how they circle about around a structure that eventually leads into a duct in order to send off this exocrine secretion. And this exocrine secretion is filled with several types of enzymes that can break down our carbohydrates, our proteins, our lipids, and our nucleic acids. So all four of the major groups of macromolecules can be broken down by the exocrine secretions of these pancreatic acini. So do notice that they're all going to be secreting and sending their contents out through these ducts to eventually make their way to the major pancreatic duct, which is going right down the middle of this pancreas. And notice that here you have the common bile duct, bringing that bile down for the emulsification of fats. So that's coming down this way. And then you have this enlargement known as the ampulla of Vater, where the two ducts meet. And finally, and we've seen this already, you've got that major duodenal papilla, which is where the mixture of bile and pancreatic juice can squeeze out into that C-shaped duodenum. Now, in this image, we get an interesting view of how we can control the secretions here. Notice that you've got sphincters that are closing off these tubes in case we don't need them. So when we're not digesting food, we don't want these contents going into the duodenum. So notice you've got the pancreatic duct and sphincter. So that circular structure going around that duct right there is the sphincter, as well as the bile duct and sphincter. And you also have the sphincter going around that ampulla. Over here we can see how that would work. A shows us when the sphincter is relaxed, and we can see that ampulla of Vater open, allowing for a droplet of that mixture of bile and pancreatic juice to basically squirt out of that papilla. In B, on the other hand, you can see that when the sphincter is closed, you don't have as much of the secretion coming out of that papilla. Down here, what is interesting in this image is that we've got the sphincter being shown to us there. We've got the ampulla with the papilla there, the ampulla of Vater and that major papilla. And we see that in this image, they have lodged a gallstone in that ampulla, closing off that duct, giving us a glimpse into the problems that gallstones can give us when they're lodged inside these ducts, not allowing those secretions to come out. As a reference, up here you can see the common bile duct and the major pancreatic duct. Notice that the major pancreatic duct is also known as the duct of Wirsung. The major pancreatic duct would therefore be this one. All of that is known as the duct of Wirsung. And compare that to the accessory pancreatic duct, which we mentioned is not found in all individuals and is there by itself without the bile duct. And that one is also known as the duct of Santorini. Now, an interesting note about the name Wirsung. Here we have the original picture, a drawing of the duct when it was first discovered. This is drawn by Wirsung himself. And you can see that pancreas right there with that duct going down the middle. The reason why we call it the duct of Wirsung is because he is the one that we believe discovered that duct for the first time. However, there's a very interesting story behind this, as is being shown by this drawing over here. Let's read some history. This was taken from an article known as Death and the Doctor by Sanjay A. Pai. And it says, poor Johann Wirsung, professor of anatomy in Padua, Italy, was murdered while entering his house at night in 1643, probably as a result of a quarrel over who was the first to discover a duct. He was killed by a Belgian student named Giacomo Cambier 1 year after he discovered the duct. The duct, whose function was never known to Wirsung, in other words, he didn't know what it was for, he just saw it in a dissection and drew it, was eventually named after him and is now termed the duct of Wirsung, or the main/major pancreatic duct. So here is Giacomo Cambier shooting Wirsung over a scientific dispute. Now, it is interesting to note this, not only because we see that his name is attached to this duct, but also because we see how anatomy, the way we study it today, has a history behind it of discovery and sometimes humans fighting each other over whose name gets to go inside textbooks. Luckily nowadays, we don't name new discoveries so much after somebody's name, rather we use the function of the structure for the name. And hopefully this little story will help you to never forget the pancreatic duct of Wirsung. Let's move on and look at some of the anatomy and the physiology of the stomach. The first thing to note is that, as we mentioned before, the stomach will receive that bolus from the esophagus as it's moving down. And once it enters the stomach is a location of extreme mechanical and chemical digestion. The mechanical is from gastric churning, as has been mentioned already. And the way that this works is through those 3 layers of muscle. We've got the longitudinal layer, the circular layer, and the oblique. Notice the direction of the fibers, right? The longitudinal is going long ways, the circular is going perpendicular across, and the oblique is going at an angle. So these 3 layers of muscles are going to squeeze on that food and break it down mechanically. But you also want to notice that the stomach is extremely folded, both macroscopically with those rugae, but also microscopically. If we take a little piece of that ruga and zoom into it under a microscope, we see that it is extremely folded with these gastric pits and these gastric secretory portions made up of different types of cells. We have the mucus cells, which make a lot of mucus to protect that stomach lining. We also have parietal and chief cells, which are going to be creating a very acidic environment and an environment for protein enzyme digestion. And that's where your chemical digestion will come from. And we also have an endocrine component with the G cells for communication that takes place throughout the digestive system through hormones that we're going to speak about shortly. Let's look at the histology of this slice of stomach tissue. Notice that we have the mucosa, which looks extremely folded, increasing the surface area. In this case, that surface area is large in order to fit in as many gastric glands as possible. And that's going to be for chemical digestion using enzymes. You can see the entry points known as gastric pits. And underneath that mucosa, you have the muscularis, which is a thin muscle right under that mucosa. And finally, the submucosa. We're going to pay special attention to the gastric glands. So let's take a look at what they are like and what their cells are like. Let's notice first the gastric pits. You can see that these are them, these openings, which lead you down into these tunnel-shaped gastric glands. And within those gastric glands, you have mucus cells towards the top. And below that, you have chief cells and parietal cells. Chief cells being in higher numbers, that's why they're called chief cells, and parietal cells being fewer and in between. Let's see what contents they are responsible for. The parietal cells are making hydrochloric acid. Notice that you have Cl- and H+ coming out of them. And these get together to make a very potent acid known as hydrochloric acid. It is important to note that the pH found in the stomach is roughly a pH of 2, which is extremely acidic. This type of pH is important because it basically burns right through tissues, and it is responsible for denaturing or unfolding the proteins that we consume. This acidic pH is such a acidic environment that it can actually start to burn right through that epithelium of the stomach itself. And that is why it is extremely important that the stomach lining makes an incredible amount of mucus as a protective barrier against that hydrochloric acid, so that the hydrochloric acid does not begin to burn holes through that epithelium. Which does, by the way, happen, and we use the word ulcer as a word to describe when we have injuries to that stomach lining, which can be extremely painful. The chief cells, on the other hand, are the cells that are making the enzyme to break down protein. But notice that chief cells make a precursor to the enzyme, known as pepsinogen, which is an inactive enzyme. However, at this stage, you can see that the hydrochloric acid converts the pepsinogen into an active form of the enzyme known as pepsin. In this image, we can get a better understanding of how that works. Note that we have the gastric gland, and we have labeled for us the chief cells and the parietal cells. The chief cells, again, are making pepsinogen. Please note that it has these amino acids that are in purple, but also a string of amino acids in orange. And it is the orange ones that make it inactive. It is the orange ones that have to be removed in order to activate it to pepsin. And that's exactly what hydrochloric acid can do. So once the hydrochloric acid comes out of the parietal cells, it will meet the pepsinogen and cleave or cut that orange peptide, giving us our product, pepsin, which is an active enzyme. Now, please remember that enzymes are, in fact, proteins that your body makes. And in this case, this enzyme is breaking down the proteins that we're eating from our food. So imagine that you just ate a sandwich and that sandwich has protein in it. That protein ends up going into the stomach, and this protein needs to be broken down further by cutting through those peptide bonds. And that's what pepsin can do. Pepsin will go and start to cut up the dietary proteins into smaller bits of amino acids. And that is, of course, a perfect example of chemical digestion, taking a large molecule of protein and slicing it into smaller pieces of protein. Let's talk about the endocrine component of the digestive system in this part of the body. Please note that we've gone over some of this already. We have parietal cells that are making our hydrochloric acid. We have chief cells that are making our pepsinogen. We have mucus cells that, of course, make mucus. But we want to focus on this piece right here. We want to focus on G cells in this case, which are endocrine cells that make an enzyme known as gastrin. If it is, a hormone known as gastrin. If it is a hormone, we should expect that it goes into circulation, which it does. In other words, it goes into the bloodstream, right? And once it's inside the bloodstream, the question is, what does it do? Well, what's interesting about gastrin is that it actually feeds back onto the stomach itself to increase the secretion of those gastric glands to make even more gastric juice. In other words, it's a mechanism to increase gastric motility and secretion. What could cause gastrin to be secreted by these G cells, these endocrine cells? Gastrin is released in response to stomach distension, which just means the stretching of the stomach. And the way that that would happen is if food went into the stomach and caused the stomach to be stretched out. Another factor that could cause this is an increase in the pH of the stomach. We want the pH of the stomach to be low. So if the stomach pH goes up, the G cells can respond to make gastrin, which would feed back towards those parietal cells and those chief cells and tell them to make more of that gastric juice to bring that pH back down. Let's take a bigger view of this to see how it could work. Notice that the brain is a big part of this as well. The taste, the smell, or even the thought of food, or tactile sensation of food in the mouth, in other words, feeling food in the mouth, will stimulate the brain. The brain, in turn, will send signals via the parasympathetic portion of the nervous system with the vagus nerve towards the enteric plexus, which is the nervous system of the digestive system in the gut. And notice that it says that this will stimulate gastrin by endocrine cells. Gastrin is carried through the circulation back towards the stomach, where it stimulates secretion. And that's what's being shown here. We get stimulation of these G cells. They make the gastrin. The gastrin goes into circulation and feeds back onto the stomach so that the stomach can increase its motility, its mechanical digestion, in other words, as well as its gastric juice secretion for the chemical digestion portion of it. If we continue, we see that we have the phase when food actually enters the stomach, known as the gastric phase. So here we want to think about food actually going into the stomach. And at this point, we can see that that would cause distension or stretching of the stomach. Distension of the stomach will stimulate mechanoreceptors, stretch receptors, and activate a parasympathetic reflex. This will cause further secretion of the endocrine cells. Gastrin is secreted, is carried through the circulation back to the stomach, where it will stimulate further secretion of those gastric juices. The next phase is when food goes from the stomach and starts to enter the duodenum. We call this phase the intestinal phase. The material that has now been digested in the stomach is known as chyme, and chyme will start to go into the duodenum. At this point, we want to slow down the gastric part of digestion. And we do this with with hormones, I should say. With hormones that are being secreted, in this case, by the duodenum. The hormones that are secreted by the duodenum are known as secretin and cholecystokinin. Notice it says that chemoreceptors in the duodenum are stimulated by this low pH from the chyme that's coming out of the stomach. And what we want to do now is decrease the gastric secretions. We want to make less of that gastric juice in order to slow down the stomach so that the stomach can start to empty little by little into the duodenum. Remember, the duodenum cannot handle a lot of that chyme coming through. It can only take it little by little. In order to achieve that, we need to slow down what the stomach is doing so that it can now empty its contents into the duodenum. Therefore, enzymes, I keep saying enzymes by mistake, I should say hormones coming out of the duodenum, secretin and cholecystokinin, go into circulation and feed back onto the stomach this time and start to shut it down. And notice it says it decreases gastric secretions in the stomach. Let's take a further view of this. Let's try to imagine this a little bit better. Here is the chyme. The stomach is filled with it now. It has done its mechanical digestion through gastric churning. It has done its chemical digestion through hydrochloric acid and pepsin. But now that material has been broken down to a soupy, acidic, nutrient-rich concoction known as chyme. We now have to move into the intestinal phase. Notice what is stopping that chyme from entering the duodenum is a sphincter known as a pyloric sphincter. It is closed off because the duodenum can get burnt from this very low pH of chyme. When the pyloric sphincter does open, it will open little by little. We will see peristaltic waves, and these waves will allow a little bit of the chyme to get pushed towards the pyloric region, and some of these peristaltic contractions will force a few millimeters of the fluid chyme through the pyloric opening into the duodenum, as is being shown right here. When that happens, the duodenum receives this very low pH, and in order to prevent itself from being injured, sends hormones, secretin, and cholecystokinin, or CCK, towards the stomach, telling it to slow down. We can see that very clearly in this image. We can start all the way up here from the liver. We see that bile coming down into the gallbladder. And then eventually coming down, we see that we have that pancreatic juice coming from the pancreas. We've seen all this before. And now we can also add that we have that chyme that is coming down through that duodenum. That duodenum realizes that the chyme is in there, that it's extremely acidic, and therefore it wants to shut down the gastric motility, but it also wants to increase, as you can see with this plus sign, CCK plus means cholecystokinin is going to stimulate the gallbladder to release that bile. It will also stimulate the pancreas to release that pancreatic juice full of enzymes. Secretin will do its part as well to get that pancreatic juice, more specifically to release bicarbonate. Bicarbonate, or HCO3 minus, is a buffer. Remember, the chyme is extremely acidic. That bicarb buffer is going to come in through that pancreatic juice and neutralize that chyme in order to protect the duodenum so we don't burn through the tissue of the duodenum. So now, hopefully, we have seen the entire picture of this portion of the anatomy that we want to go over. As a quick review, the digestive system does continue. It's got the jejunum and the ileum of the small intestine. It also has the large intestine, which does its part. But today we focused in what is in this box. And so after this lecture, you should be familiar with the functions of the liver when it comes to digestion, the gallbladder, the pancreas, the stomach, and the duodenum. You should have an understanding of mechanical digestion as well as chemical digestion, and the hormones that are being released by the stomach and the duodenum in order to regulate digestion in this particular area. As a last exercise, I would like to look at this chart in order for us to better understand what chemical digestion is like, and specifically to focus on how rich pancreatic juice is in terms of the enzymes it has. So notice that we have the enzyme name, the source, what it breaks down, and notice that the pancreas, those acini, those exocrine acini, are responsible for making a big portion of this chart. Carboxypeptidase is important for breaking down peptides, in other words, protein. Chymotrypsin, also for protein. Deoxyribonuclease to break down DNA and nucleic acid. Elastase, further breakdown of proteins. Pancreatic amylase to break down starches, which are carbohydrates. Pancreatic lipase to break down triglycerides or fats. Ribonuclease to break down RNA. Trypsin to break down proteins further. So pancreatic juice, going through that duct of Wirsung, and remember that Wirsung, when he died, he did not know what was going through that duct, but he does have his name attached to it. These are the enzymes that are going through it. We can compare that to, for example, what is coming out of saliva. You've got salivary amylase and salivary glands that break down starches. You've got lingual lipase, lingual glands and tongue for triglycerides, fats, in other words. And in the stomach, we talked quite a bit about pepsin, which is to break down proteins, but there's also gastric lipase to break down triglycerides. Further down in the small intestines, in the jejunum and then the ileum, you have more enzymes to break down these contents into smaller bits so that they can eventually be absorbed towards the end of the small intestine. And that absorption is, of course, important to get those nutrients into the bloodstream.